Hi friend, welcome to PLANTI.com. Today I am going to demonstrate the denaturation of protein in biochemistry. At first, denaturation of protein. Denaturation, that means nature change of protein. Denaturation of the protein is a condition when the unique three-dimensional structure of a protein is exposed to changes. This is three-dimensional structure. This is exposed to changes. Due to changes in how change it? Due to temperature, pH or other chemical activities. The hydrogen bonds present in the proteins get disrupted, and this protein have so many hydrogen bonds that disrupted. This results the unfolding of globular protein, and when the hydrogen bond and other so many bonds are disrupted, it causes unfolded protein. This is properly folded protein, and when the hydrogen bond and other bonds are disrupted, then it unfolded protein and uncoiling of the helix structure. It is properly coiled. Here you see it is properly coiled, but this protein uncoiling of helix structure. This helix is not properly coiled, unfolded protein. Next, the uncoiling of helix structure affects the chemistry of proteins and they lose their biological activity. That means if a properly folded protein loses its structure and become unfolded, it also loses its biological activity. This phenomenon of losing their activity and uncoiling of helix structure due to physical or chemical changes is called the denaturation of protein. That means suppose you have this 8 amino acid sequence, both are connected by peptide bond. Suppose it is properly folded in this way. So, when suppose the heat present and it disrupted and it is in this way present. So, here suppose it work good role in biological activity, but it is not. Though 8 amino acids are present in both cases, but due to folding difference, these protein are different. This folding is very important. And the bond are helping proper folding. During denaturation of proteins, the secondary and tertiary structure get destroyed and only the primary structure is retained. Here you see that I say here 8 primary 8 primary amino acid are present, primary structure, and here secondary structure disrupted and tertiary structure also disrupted. So when it called denaturation of protein, that does not mean the primary structure disrupted. Primary structure never disrupted. Only secondary, tertiary and quaternary structure disrupted. When primary structure disrupted, this called damage of protein. or destroy of protein. That means primary structure disrupted. When all protein are destroyed, damaged, then primary structure disrupted. But denaturation, that does not mean primary structure disrupted. It only means secondary, tertiary and quaternary structure disrupted. Obviously, secondary structure and tertiary structure disrupted means quaternary structure also disrupted. Covalent bonds are broken and interaction between amino acid changes get disrupted. Covalent bond also disrupted, hydrogen bond disrupted. Covalent bonds are broken and interaction between amino acid chains gets disrupted. This result in the loss of biological activity of the protein. This result, hydrogen bond and covalent bond all are when disrupted, then the result, the loss of biological activity. When proper folding is disrupted because the bond is disrupted, then the biological activity also disrupted. Example of denaturing agents. At first, heat. Heating, exposure to acids or bases and even violent physical action can cause denaturation to occur. As the temperature increases, the rate of molecular vibration increases. Eventually, weak interaction such as hydrogen bonds and van der Waals interactions are disrupted and the protein unfolds. When a protein, properly folded protein, and you put heat here, so it is unfolded because the van der Waals interaction, hydrogen bonds all are disrupted and this protein is unfolded. Example, the albumin protein in egg. In egg, albumin protein present. In white part, albumin protein present is denatured by heating. When you put heat, it is denatured so that it forms a semi-solid mass, and the liquid white part becomes semi-solid mass. Almost the same thing is accomplished by the violent physical action of an egg beater in the preparation of meringue. That means a egg, when the white part of egg is liquid, when you put heat here, the white part is semi-solid condition, it gets semi-solid condition. That is cause of denaturation. The protein is unfolded and it is become semi-solid. Heavy metals. Second, heavy metals. Heavy metals poisons such as lead, cadmium. Lead, cadmium is a heavy metal, changes the structure of proteins by binding to functional groups on the protein surface. They may disrupt salt bridge by forming ionic bonds with negatively charged group. Heavy metals also bond with sulfhydryl group. Here heavy metal disrupt salt bridge and forming ionic bond form, and heavy metal also bond with sulfhydryl group. Heavy metal, strong acid or bases. Changes in pH result in protonation or deprotonation. pH change, protonation, it may be deprotonation. The side group of amino acid of protein which alters hydrogen bonding and salt bridge patterns. If a protein properly folded here, you put strong acid or base. So it can be protonation or deprotonation due to R group, side group, and it alters hydrogen bonding. If pH change, it alters hydrogen bond and salt bridge pattern. That's why the proper folding is disrupted and it is unfolded. Organic solvent, here also same in case reagents such as ethanol, organic solvent, example ethanol, that are capable of forming intermolecular hydrogen bonds with protein molecules disrupt the intermolecular hydrogen bonds within the molecule. Organic solvent, that means ethanol, suppose a protein here also properly folded, you put here ethanol and it form capable of forming intermolecular hydrogen bonds. It form hydrogen bond and here other hydrogen bond, and it also may disrupt it. The proper folded structure is disrupted by if you put ethanol in the intermolecular hydrogen bond within the molecule. Okay. Detergents are amphipathic molecule which disrupt hydrophobic interaction and unfold proteins into extended polypeptide chain. Detergent, here a folded protein here suppose suppose in this way, here present in this is hydrophobic region and this is hydrophilic region. The detergent make break this hydrophobic region and the proper fold is disrupted and it is unfolded protein. So here I want to say that a properly folded protein here, suppose you put detergent here, then it is unfolded polypeptide chain. Reducing agent such as beta markovtethanol reduce disulfide bond to sulfhydryl group and break intra or interchain disulfide bond. This beta markovtethanol play a vital role in breaking disulfide bond. Suppose here sulfur bond here, if you put a beta markovtethanol here, then the sulfhydryl bond break here. In this case, the properly folded protein here, if you put beta markovtethanol, then this is misfolded because beta markovtethanol break all disulfide bond. This may be intra or inter. All disulfide bond are break, then it is unfolded. So the secondary tertiary structure are disrupted and only polypeptide chain are remain. This is primary structure. This is the whole concept of denaturation. And I want to say a small thing, renaturation of protein. What is a renaturation of protein? Most of the denaturation process are irreversible. Most of case, if a properly folded protein, you put heat, heavy metal, pH change, acid or base, and detergent, reducing agent, if you put here, then it is unfolded, and most of the cases it can't properly folded. Again it not properly folded. but in some case it also be properly folded. This case, but it has been seen in very few cases that some of the denaturation process can be reversed. It is then called renaturation of protein. Here the image, it is normally properly folded protein. If denature, you put here all denaturing agent, heat, then the hydrogen bond, covalent bond, hydrophobic bond, disulfide bond, all are break and denaturation of protein here. In this case, mainly it never form properly folded protein, but in some case, in very few cases, it can be properly folded. This is called renaturation. Denaturation means a properly folded protein become unfolded, and when unfolded protein become folded, this is called renaturation. Thank you for watching my video. I think this video going to helpful for you.